Hello, I'm Professor Sims, and in this video I will discuss the basics of genetics and inheritance. This is the 6th in the series of 10 lessons held as part of my Gen Bio 1 course. If you are a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include describing behavior of chromosomes and cellular events during meiosis. We'll talk about the differences between meiosis and mitosis. We'll explain the mechanisms within mitotic processes that produce genetic variation. We'll identify Mendelian and non-Mendelian inheritance patterns. We'll talk about Sutton's chromosomal theory of inheritance, genetic linkage, homologous recombination. We'll talk about chromosome creation, the 3-point test crosses, karyograms, and errors in chromosome structures. Reproduction is a fundamental aspect of life. Some examples that the book gives are hippopotamuses that give birth to calves, the Joshua trees that produce seeds that grow into seedlings, and then they talk about flamingos that lay eggs that hatch into chicks. And they give these examples because it's many kind of different ways that sexual reproduction is happening in different types of organisms. While some organisms can reproduce asexually, asexual reproduction is where they create genetically identical clones, right? Well, many organisms, both single-celled and multicellular, rely on sexual reproduction. And sexual reproduction involves the production of two distinct haploid cells by the parents, which then fuse to form a genetically unique diploid cell, a new organism. These haploid cells are commonly referred to as male and female, and they're produced through meiosis, which is similar to mitosis, but we'll talk very explicitly about the differences. Sexual reproduction, with its combination of meiosis and fertilization, introduces variation into the offspring, and this contributes to the success of sexual reproduction. The majority of eukaryotic organisms, including plants and animals, rely on meiosis and fertilization for reproduction. Sexual reproduction involves the fusion of two specialized cells called gametes. Each contain a set of chromosomes, and when these gametes unite, they form a fertilized egg or a zygote that has two sets of chromosomes. To prevent a doubling of chromosome sets within each round of fertilization, the diploid cell must be reduced in half. So it wants to cut its chromosome number in half, and then this reduction is achieved through meiosis, and then once the gametes are fertilized, it restores the diploid chromosome number. Here is a brief overview of meiosis. It's very similar to mitosis, but it does involve two rounds. So you have cell division that's occurring in sexually reproducing organisms. It involves two rounds of division known as meiosis 1 and meiosis 2. The purpose of meiosis is to reduce the chromosome number by half, resulting in the formation of haploid cells. It occurs in specialized cells called germ cells or gamete. During meiosis, DNA replication takes place before the start of meiosis 1. Meiosis 1 involves the pairing and crossing over of homologous chromosomes, and that leads to genetic recombination and increased genetic diversity. Homologous chromosomes separate during meiosis 1, and that results in two haploid daughter cells with duplicated chromosomes. And then meiosis 2 involves the separation of the sister chromatids, and then the result of meiosis 1 and 2 is the production of 4 genetically distinct haploid cells known as gametes. And then these gametes fuse with other gametes during fertilization, and that restores the diploid chromosome number in the offspring. So that's what it looks like overall. Right? The starting nucleus is diploid. It goes through one round of chromosome replication followed by 2 rounds of nuclear division. And that's how the haploid daughter cell nuclei are produced. Before I break down each individual step,
many organisms pass their genes to their offspring through sexual reproduction. This begins when two gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information on to their own offspring. Gametes are formed through a process called meiosis. The cells that undergo meiosis to produce the gametes are called germline cells. In diploid organisms, germline cells have 2 copies of each chromosome. Germline cells undergo meiosis to produce haploid gametes, which only have 1 copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult. Meiosis is just one step in the life cycle of a germline cell. Similar to mitosis, the cells also pass through the interphase, G1, S, and G2 stages before they enter meiosis. The DNA inside a germline cell is duplicated before meiosis begins during the S phase. The duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are 2 cell division events during meiosis. The first division, meiosis 1, results in 2 unique daughter cells that have half the amount of DNA as the parent germline cell. The second division, meiosis 2, results in 4 unique haploid cells that only have 1 copy of each chromosome. These haploid cells are the gametes that could go on to produce an offspring through sexual reproduction. Let's look more closely at each of the division events. Meiosis begins with prophase 1. In this stage, the DNA condenses to form chromosomes. Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout meiosis 1. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving 2 pairs of sister chromatids. Chromosomal material is exchanged between the 2 pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no 2 siblings, aside from twins, are genetically identical. There are several more key steps in prophase 1. The nuclear membrane begins to break down. Then, the 2 centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis 1 is called metaphase 1. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is anaphase 1. During this phase, homologous chromosomes separate and migrate to the 2 poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis 1 are telophase 1 and cytokinesis. Here, the cell divides into 2 daughter cells. Each of these 2 cells now undergo meiosis 2. Meiosis 2 is similar to mitosis. The first stage of meiosis 2 is prophase 2. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase 2 and prophase 1 is the fact that the daughter cells have only 1 copy of each homologous chromosome. So in prophase 2, there is no synapsis of homologous chromosomes or crossing over. In metaphase 2, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In anaphase 2, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase 2, the nuclear membrane reforms and the cytoplasm is divided into the 2 haploid daughter cells. This division is called cytokinesis. Since meiosis 2 began with 2 cells, and each of those cells were split into 2 cells, we now have 4 unique haploid cells at the end of meiosis. These cells are gametes. 2 gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.
Figure 11.4 shows how genetic material is exchanged during recombination, or also known as crossing over in prophase 1. So we have homologous chromosomes that are lined, and then notice that these are not sister chromatins. All of the red, these are sisters to each other. The blue, those are sisters to each other. So these are homologous. They're genetically identical. But when they are lined with chromosomes that are not genetically identical, what happens a lot during prophase 1 is that those chromosomes overlap to the point where they actually are exchanging parts of their DNA, and then they are recombined into new chromatids. So these are recombinant chromatids, this one, and then these ones here are non-recombinant. So now you have non-homologous chromosomes, non-identical chromatids. Figure 11.5 illustrates the random assortment that occurs during meiosis 1. So you see here, we've got AA chromosome, BB, BB, right? Then because of the way that these get separated during meiosis, during metaphase 1, you can have 4 gametes. You may end up with capital A, capital B. You might have lowercase a, lowercase b, right? But you can also end up with capital A, lowercase b, capital A, lowercase c, and then lowercase a, capital B. And this all just has to do with how the chromosomes line up during metaphase.
When I was first learning about mitosis and meiosis, I learned them both separately first. And then I tried to figure out what was the same about them, what was different, why did they both matter. I would try to compare the stages by flipping through images. You know what would have really helped me? A side-by-side comparison. And that's what this video is. We assume you already have a background of mitosis and meiosis. If not, take a look at our videos on them. But this video is a side-by-side comparison, presented in a split screen. Mitosis on the left, meiosis on the right. Both of these processes, along with the cytokinesis that follows them to split the cytoplasm, are involved in making new cells. Mitosis results in body cells. Meiosis results in sperm and egg cells, otherwise known as the fancy term gametes. Before we start mitosis and meiosis, let's look at what you start with. Your starting cell in both mitosis and meiosis is diploid, written here as 2N. That means it has 2 sets of chromosomes. In humans, that's including 1 set of 23 chromosomes from mom and 1 set of 23 chromosomes from dad. 46 chromosomes total. During interphase, the cell duplicates the chromosomes. When you duplicate 46 chromosomes, you still say there are 46 chromosomes, as the newly duplicated portion is still attached at the centromere region. But there are actually 92 chromatids. Interphase isn't part of mitosis or meiosis, but it's a really important phase because it duplicates chromosomes before we get started. Just to point out, it's really hard to draw 46 chromosomes. We're going to use 6 chromosomes in our diagrams when we illustrate what's happening, because it's much easier to draw and visualize. Oh, and just a fun fact, some insects have 6 chromosomes, like mosquitoes. Unfortunately, I'm not a fan of mosquitoes, but mosquitoes do both mitosis and meiosis. When learning the stages, we give the acronym PMAT, which is helpful for understanding the stages. Both mitosis and meiosis go through these stages, but meiosis goes through them twice, and therefore has a number next to each PMAT stage. We're going to show some basic events for each PMAT stage, but please know that there is way more detail to explore than what we can include in this quick video. Prophase in mitosis. Remember that pro can mean before, and this stage comes before the others. The chromosomes are visible. We say they're condensing, which means they are thickening. Prophase 1 in meiosis. Happening here too, but the chromosomes are actually going to match up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. With each pair, one came from mom and one came from dad. In this formation, chromosomes can transfer their genetic information and exchange it between each other. It's called crossing over. It can make for what we call recombinant chromosomes. Metaphase in mitosis. The nuclear envelope which had surrounded the nucleus was already disassembled before metaphase even started. For metaphase, I like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell in a single file line. Metaphase 1 in meiosis. The chromosomes are in the middle as well, but they're still going to be in pairs in the middle of the cell, so it's not a single file line. Anaphase in mitosis. I like to think as the A for away, the chromatids are pulled away by the work of the spindles. They are moving to opposite sides of the cell. Anaphase 1 in meiosis. Same thing, but in this case, it's the chromosomes, not chromatids, being pulled away to opposite sides of the cell. Telophase in mitosis and telophase 1 in meiosis. The chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make two new cells. And they are starting to surround the chromosomes on both sides, as this will eventually form two cells. Cytokinesis follows to split the cytoplasm to complete the actual dividing of the cell. So at the end of mitosis and cytokinesis, we end with two identical diploid cells. In humans, they would both have 46 chromosomes. This is great for organism growth. Growing requires making more cells, after all, or replacing damaged cells. On to meiosis 2. Prophase 2. Chromosomes are condensing in both cells. It's not going to be as eventful as it was in prophase 1 because they're not going to have homologous pairs and crossing over. Metaphase 2. M for middle, but this time the chromosomes are in a single file line, similar to how metaphase looked in mitosis. Anaphase 2. Think A for away. This time, though, it's actually the chromatids that are getting pulled away. Telophase 2. Chromosomes are at the complete opposite ends, and new nuclei are forming on each side to make these new cells. Cytokinesis will follow meiosis 2 to completely split the cytoplasm. We are now finished with meiosis, and we end with 4 non-identical cells, gametes. Males make sperm cells in meiosis, and females make egg cells in meiosis. These gametes are haploid, meaning they have half the number of chromosomes as the original starting cell. In the case of humans, the resulting cells would each have 23 chromosomes. By the way, when a sperm and egg cell combine, it results into a diploid cell, a fertilized egg, otherwise known as a zygote, which will then start a series of divisions using mitosis to give rise to a brand new organism.
So like we saw in the video, meiosis and mitosis are both processes involved in cell division, but they differ in their purpose, in their stages, and in their outcomes. Firstly, meiosis occurs in specialized cells called germ cells, and the main goal is to produce gametes with half the number of chromosomes as the parent cell. In contrast, mitosis occurs in somatic cells and is responsible for growth, repair, and asexual reproduction. So mitosis is making two genetically identical daughter cells. Here you can really see the difference. Where meiosis has 2 rounds of division, mitosis only has 1. Meiosis 1 involves making homologous chromosomes, pairing up, crossing over, separating, and leading to genetic diversity. Meiosis 2 separates the sister chromatids, making 4 haploid daughter cells. In mitosis, the sister chromatids are separating during anaphase, and it makes 2 diploid daughter cells. Genetics is the study of heredity. Johann Gregor Mendel laid the foundation for genetics long before the discovery of chromosomes or genes, when meiosis was poorly understood. Mendel conducted meticulous quantitative analysis on a simple biological system with large sample sizes, revealing the fundamental principles of heredity. We now know that genes carried on the chromosome are the functional units of heredity responsible for replication, expression, and mutation. Mendel's postulates form the basis of classical genetics, although not all traits follow Mendelian patterns. His experiments provide an excellent starting point for understanding inheritance. For his experiments, because it naturally self-fertilizes and it produces true breeding plants. So this allows for him to avoid unexpected traits popping up in the offspring. The garden pea also has a short life cycle, so that allows for evaluation of multiple generations within a short period of time. And then large quantities of pea plants can be cultivated at the same time, so that reduces the role of chance affecting the results because you have a large sample size. In his crosses, Mendel mated true breeding individuals with different traits, and this is known as hybridization. He manually transferred pollen from one plant to another plant in order to prevent self-fertilization, and then the offspring of these crosses were called the F1 generation. And then Mendel allowed the F1 generation to self-fertilize naturally, and that produced the F2 generation. And Mendel observed a 3-to-1 ratio of dominant to recessive traits in the F2 generation. Chromosomes determine physical characteristics, and peas, like any other plant or animal, are diploid organisms with 2 copies of each chromosome. These copies have the same gene order, and then during fertilization, haploid gametes unite to create a diploid zygote. In diploid organisms, each gene can have 2 different versions called alleles. Alleles are found at the same location on homologous chromosomes, and then they have some variation that has arisen from mutation. So it's common to find more than 2 alleles for a gene in a natural population. The physical traits that are expressed by an organism are called its phenotype, while its underlying genetic makeup is called the genotype. And Mendel's experiments with hybridization showed the difference between the phenotype and the genotype. F1 heterozygous offspring resemble one parent due to the dominant and recessive alleles. So Mendel called dominant traits those that were inherited unchanged in hybridization, and then recessive traits were latent and then reappeared in subsequent generations. So Mendel proposed that plants possess 2 copies of each trait, and each parent contributes 1 copy to their offspring. Mendel's findings, based on the principles of probability, revealed the basics of heredity because he was able to demonstrate that traits were transmitted as discrete units. They were independent of each other. And then by applying the product and some rules of probability, Mendel was able to predict the outcomes of the crosses. When true breeding plants with yellow and green pods were crossed, the hybrid offspring all had yellow pods. However, when the green pod allele reappeared, it was in later generations, indicating that there was a difference in the genotypes. The parent plants were homozygous for the traits that he studied, meaning that they had 2 identical alleles for the gene. When contrasting traits were cross-fertilized, the offspring were heterozygous, having different alleles for the genes. To illustrate a monohybrid cross, let's consider true breeding pea plants with yellow and green seeds. The dominant seed color is yellow, so the parental genotypes are Y for yellow-seeded plants and lowercase y for green-seeded plants. We can use a Punnett square created by Reginald Punnett to predict possible outcomes of a gamete cross and their expected frequencies. The Punnett square lists all possible combinations of parental alleles and shows the resulting diploid genotypes of the zygotes. From the Punnett square, genotypic and phenotypic ratios can be determined. For a monohybrid cross with true breeding parents, there's only one possible genotype, which is uppercase Y, lowercase y, resulting in all offspring having yellow seeds. If we do a self-cross of one of the heterozygous offspring, we can use a 2x2 Punnett square because each parent can donate one of the 2 different alleles. This results in 4 possible allele combinations. You have Y, lowercase y, capital Y, or lowercase y. And it's important to count both possibilities of obtaining the heterozygous genotype, a capital Y from the egg and a lowercase y from the sperm, or a lowercase y from the egg and a capital Y from the sperm, right? So that's where the difference is where they're getting them from. And despite their alleles coming from different parents, both combinations yield offspring that are genotypically and phenotypically identical. Since fertilization is random, each combination is equally likely, leading to a genetic ratio of 1 to 2 to 1, heterozygotes being the 2, right? So the ratio is 1 homozygote dominant, 2 heterozygote, and 1 homozygote recessive. And then Mendel confirmed these results by doing an F3 cross, which is self-crossing the F2 plants. And when the green-seeded plants were self-crossed, all the offspring had green seeds, confirming that green green seeds have the homozygous genotype lowercase y. In the self-cross of the yellow-seeded plants, 1/3 of the plants had yellow seeds, while 2/3 exhibited a 3-to-1 ratio of yellow to green. So the true breeding plants had the homozygous genotype lowercase y, and the segregating plants had the heterozygous genotype. Self-fertilization of these plants produced the same results as the F1 self-fertilizing cross. Here's another way to look at it. So this is what is called a test cross. Mendel developed the test cross to determine if an organism is expressing a dominant trait because it's a heterozygote or is because it's a homozygote. So in a test cross, the dominant expressing organism is crossed with a homozygous recessive organism for the same trait. If the dominant expressing organism is a homozygote, all the F1 offspring will be heterozygotes expressing the dominant trait. Alternatively, if the dominant expressing organism is a heterozygote, the F1 offspring will have a 1-to-1 ratio of heterozygotes and recessive homozygotes. The test cross confirms Mendel's idea that unit factors segregate equally. Let's talk about alternatives to dominance and recessiveness. Mendel's pea plant experiment showed that genes come in pairs, with each parent contributing one allele. The dominant allele takes precedence, hiding the recessive one and not contributing to the phenotype. So recessive alleles can be carried without being expressed. And individuals with one dominant and one recessive allele are sometimes called carriers. Further studies in other plants and animals have revealed more complexity, but the basic principles of Mendelian genetics still hold true. One extension of Mendel's ideas is incomplete dominance. Sometimes the phenotype of heterozygotes appears to be intermediate between the parents. For example, in snapdragons, crossing a homozygous white-flowered parent with a homozygous red-flowered parent produces offspring with pink flowers, and this is known as incomplete dominance. In this case, the genotypic ratio would be 1:2:1, and the phenotypic ratio would also be 1:2:1. In codominance, you have both alleles simultaneously expressed in the heterozygote. So an example of this would be blood groups in humans. You have the M and N alleles, and they're expressed as M or N antigens on red blood cells. And then homozygotes express only one allele, while heterozygotes express both equally. Mendel suggested that only 2 alleles, one dominant and one recessive, exist for a given gene. But we now know that this is an oversimplification. Multiple alleles can exist at the population level, resulting in many combinations. And the most common phenotype or genotype among wild animals, this is designated as the wild type. Multiple alleles can exhibit dominance hierarchies. For example, in rabbits, there's 4 alleles for coat color, with the wild type allele expressing brown fur, the chinchilla allele expressing black-tipped white fur, and the Himalayan allele expressing black fur on the extremities, and the albino allele expressing white fur. So this is an example of a trait that has multiple alleles. And notice that there's not a huge difference in the ratios. It's just that these intermediate genotypes exist. And the result is not just brown or white fur. It's brown, white with black on the tips, white with black on the extremities, or just white. In sex-linked traits, the sex of an individual is determined by sex chromosomes. Humans have 22 pair of autosomes and then just 1 pair of sex chromosomes. Females have 2 X chromosomes, while males have 1 X and 1 Y chromosome. And then there is this phenomenon called hemizygous, meaning that you can have only 1 allele for X-linked characteristics. And then that's generally happening in males, whereas females, they have 2 X's. Genotypes and phenotypes of the offspring in an X-linked cross depend on the expression of the recessive trait by the male or female in the P1 generation. So eye color in fruit flies is one of the first X-linked traits studied. The wild type eye color is red, and red is dominant over white. If the male expresses the white-eyed phenotype and the female is homozygous red-eyed, and that means that all of the F1 generation will have red eyes. F1 females will be heterozygous. F1 males males will all have white eyes. Let's have a look at figure 1213, and this is giving an example of female carriers and heterozygous carriers and how things are inherited differently in males and females. A male is going to be XY, female's XX. This orange color is affected, so let's say that this is hemophilia. Mom is a carrier. Dad doesn't have it at all. If mom passes on the X that has the dominant allele, so the dominant allele in this case would be not hemophilia, and she passes that on, then she'll have either a carrier or unaffected male. If she passes on the X-linked recessive allele, then she can also have a carrier or an affected male. And the dad, he can only pass on the dominant X or the Y. So he doesn't have the X-linked recessive allele at all. If any of the children have it, they're getting it from mom. I want to show you a video that goes into this process in just a little bit more detail.
Would you like to know one of the most underappreciated pieces of cytoplasm out there? Platelets. We take for granted the function of our platelets, which are fragments of cytoplasm that help stop us from bleeding. They help our blood clot when we get hurt. But there is a disorder called hemophilia that can affect those platelets because special clotting factor proteins that are needed to work with the platelets may not work correctly. So even a basic cut could be dangerous for people with hemophilia because they could bleed continuously. We have many treatments for the symptoms of hemophilia, and this has greatly improved outcomes with this disorder, although it wasn't always that way. Hemophilia is a sex-linked recessive trait, which means it's different from the basic Mendelian genetic problems. What do I mean by that? Well, we still use the terms dominant and recessive for alleles, but this time, because it's sex-linked, the alleles are on sex chromosomes. This is the case with sex-linked traits. So what is a sex chromosome? Well, recall that humans have 46 chromosomes. Chromosomes are made up of DNA and protein, and they contain your genes. Well, 2 of your 46 chromosomes, they are called the sex chromosomes. In a karyotype, it is usually the last 2 chromosomes that are the sex chromosomes. The sex chromosomes are called X and Y chromosomes, but it has nothing to do with the shape of the chromosome. That's always kind of confusing, but please don't think that Y chromosomes are shaped like a Y and X chromosomes are shaped like an X. This used to always confuse me. It has nothing to do with their name. The reason they got their name is actually kind of interesting, so to the Google for that. Everyone has an X chromosome, but if you have another X chromosome, meaning you have 2 X chromosomes, you are female. And if you have a Y chromosome, meaning you have an X and a Y chromosome, you are male. There are also genetic disorders where you can have extra copies of sex chromosomes, but we will not go into that in this clip. Sex-linked traits are traits that are specifically on the sex chromosomes. Most sex-linked traits tend to be on the X chromosome because it is larger than the Y chromosome and it contains more genes than the Y chromosome. The disorder hemophilia is like this. Now we will use the letter capital H to represent an allele for having normal blood clotting function, and a letter lowercase h to represent an allele for having hemophilia. Hemophilia is a sex-linked recessive disorder, which is why it's being represented by a lowercase letter h. Only it must be placed on the sex chromosome as a superscript, like an exponent. A woman that does not have hemophilia could have the genotype X big H, X big H, or X big H, X little h. Because as long as she's got at least one dominant allele, that dominating allele will be what shows. So no hemophilia, since again, hemophilia is a recessive sex-linked disorder. The only way for her to have hemophilia would be the genotype X little h, because only when there is no dominant present will that recessive show up, at least in this type of trait. Now, for a male to not have hemophilia, his genotype would have to be X big H, Y. Notice how I did not put anything on the Y chromosome. Again, most sex-linked traits are on the X chromosome. If he has the genotype X little h, Y, then he has hemophilia. He doesn't have 2 X chromosomes, so he cannot be a carrier. So let's say that two people that do not have hemophilia have children. However, let's say that the woman is a carrier. That means she is heterozygous. How do you do a sex-linked Punnett square cross for this kind of trait? Step 1, identify the genotypes of the parents. So the mother was a carrier. She is X big H, X little h. She does not have hemophilia because of that dominant allele present, but she is a carrier. The male, if he does not have hemophilia, well, he must be X big H, Y. There's no other option for him. Step 2, place one parent on the top, outside of the square like this, and place the other parent on the left, outside of the square like this. Step 3, cross them. For formatting purposes, place X chromosomes before Y, and you also write any sex chromosomes with dominant letters first. The results you get in the squares would be the offspring, the babies. The genotype ratio could be written out like this. The phenotype ratio, remember that these are the observable traits, can be written out that there is a 75% chance that the child will be born without hemophilia and a 25% chance that a child would have hemophilia for this boy here. You could also make this a ratio, so 3:1. Notice that in this type of example of a sex-linked recessive disorder, males are more likely to inherit this disorder because there is only one X chromosome present. This is true for many other sex-linked recessive disorders, such as colorblindness.
Mendel had 4 postulates, often called laws, from his pea plant experiments, summarizing the basis of dominant and recessive inheritance in diploid organisms. However, as you've learned, there are more complex patterns of inheritance beyond the classic 3:1 phenotypic ratios. Yet, these laws provide a foundation for classical genetics, so let's have a look. The first one is the existence of paired unit factors or genes that are transmitted faithfully throughout gametogenesis and fertilization. Contrary to the prevailing belief of parental traits blending in offspring, Mendel's experiments showed that heredity factors are discrete units. The second one, the law of dominance, states that in a heterozygote, one trait will conceal another trait for the same characteristic. Only the dominant allele is expressed, while the recessive allele remains latent but can be passed on. Offspring need 2 copies of the recessive allele to express the recessive trait. However, researchers have discovered that the law of dominance is not always applicable. Mendel proposed the law of segregation after observing true breeding pea plants with contrasting traits produced F1 generations with the dominant trait and F2 generations with a 3:1 ratio of dominant to recessive, ensuring equal chance of inheriting either factor. The F2 generation can have 3 possible genotypes: homozygous dominant, heterozygous, or homozygous recessive. Since heterozygotes can arise through different pathways, they are phenotypically identical to homozygous dominant individuals, supporting the 3:1 ratio. The physical basis for this segregation lies in the first division of meiosis, where homologous chromosomes segregate into the daughter nuclei. So let's have another look at independent assortment. According to Mendel's law of independent assortment, genes do not influence each other when it comes to sorting of alleles. Every possible combination of alleles for each gene has an equal chance of occurring. So we can understand independent assortment through a dihybrid cross, which involves 2 true breeding parents with different traits for 2 characteristics. For example, look at pea plants with green wrinkled seeds and yellow round seeds. Since both parents are homozygous, the law of segregation tells us that green wrinkled plants' gametes will be lowercase y, lowercase r, while yellow round plants' gametes will be uppercase y, uppercase r. Therefore, all offspring in the F1 generation will be uppercase y, lowercase y, uppercase r, lowercase r. So they're all going to be heterozygous for both traits. Now, in the F2 generation, the law of segregation states that each gamete must receive either a capital R allele or a lowercase r allele, along with either an uppercase y allele or a lowercase y allele. Sorting of the lowercase r allele is equally likely to be paired with an uppercase y or a lowercase y. This leads to 4 equally likely gametes when we self-cross the uppercase y, uppercase r are heterozygote. We get uppercase R, uppercase Y, uppercase R, okay? Uppercase Y, lowercase y, uppercase R, and lowercase y, lowercase y. And by arranging the gametes in a Punnett square, we obtained 16 equally likely genotypic combinations. And from these genotypes, we can infer a phenotypic ratio of 9 round and yellow to 3 round and green to 3 wrinkled and yellow to 1 wrinkled and green. And these ratios are what we would expect from a large enough sample size. Due to independent assortment and dominance, we can collapse the 9:3 to 3:1 dihybrid phenotypic ratio into 3:1 ratios, similar to what you would see for a monohybrid cross following a dominant and recessive pattern. By focusing only on seed texture or seed color, we would anticipate that 3/4 of the F2 generation will exhibit the dominant trait, while 1/4 are independent events, allowing us to apply the product rule. The physical basis for the law of independent assortment lies in meiosis 1, where homologous pairs randomly line up in orientations, and each gamete can contain any combination of parental and maternal chromosomes, the genes carried on them to this random orientation. Some combinations of alleles violate Mendel's law of independent assortment, which states that genes are inherited independently of each other, right? So we now understand that genes located on the same chromosome can be linked and tend to be inherited together. However, the process of recombination or crossover allows genes on the same chromosome to behave independently. So recombination occurs during meiosis, and segments of homologous chromosomes exchange genetic material, right? So when genes are physically close on the same chromosome, they're considered linked, and their alleles are more likely to be transmitted together. So if genes for plant height and flower color are right next to each other on a chromosome, the tall and red alleles might be inherited together, and then the short and yellow alleles are inherited separately. And this linkage affects the outcomes of dihybrid crosses because it deviates from the classical 9:3:1 ratio predicted by Mendel. As the distance between the genes increases, the likelihood of crossovers between them also increases, and this makes them behave more like genes on separate chromosomes. Geneticists use the proportion of recombinant gametes to determine the distance between genes and construct gene maps. And in reality, single observable characteristics are often controlled by multiple genes working together. Genes might act sequentially, they might complement each other, they might oppose each other to influence phenotypes. An example of this is epistasis. Epistasis is a form of gene interaction where one gene masks or interferes with the expression of another. It occurs when alleles at one locus are dependent on the function of a preceding or on a following gene in a pathway. So, for example, the presence of a separate gene is necessary for pigment production in mice, and the lack of this gene results in albinism, regardless of the allele, the coat color gene. So we're going to be talking about mostly Sutton's chromosomal theory of inheritance. But first, we got to make sure we go back and understand Mendel's chromosomal theory of inheritance. It was supported by the following observations. Okay, so you have homologous chromosome pairs migrate independently during meiosis. Sorting of chromosomes from each pair into pre-gametes appears to be a random process. Each parent produces gametes with half the chromosomal complement. The sperm and egg, despite their differences in size and shape, have the same number of chromosomes, which suggests equal genetic contribution from each parent. And the gametes combine during fertilization, which results in offspring that have the same chromosome number as their parent. With this, Gregor Mendel laid the foundation for modern genetic inheritance, but his early work was completed in the 1840s before the discovery of chromosomes under the microscope. Further, his work was met with resistance in the scientific community, mostly because there are examples in nature of blended inheritance, and because individuals have far more independently segregating traits than they had chromosomes. Advancements in microscopic techniques allowed for cell biologists to stain and visualize subcellular structures. And eventually they were able to witness the actions of chromosomes during cell division and meiosis. But before that, in 1902, we had Theodore Boveri, and he was able to determine that inheritance of chromosomes was essential for the development of sea urchin offspring. That same year, Walter Sutton was able to witness chromosome separation during meiosis, and this supported Mendel's theory of inheritance. Then we have Thomas Morgan further supported the theory through his extensive fruit fly crosses, and this provided experimental evidence in favor of the chromosomal theory of inheritance because it revealed a connection between a segregating trait and the X chromosome, disrupting Mendel's predicted outcomes. So this led to scientists realizing that genes located on the same chromosome can sometimes become unlinked during meiosis. In 1913, Eleanor Carruthers was the first person that was able to provide physical evidence. So up until now, it was just kind of mathematical, observational. She had physical evidence because she was able to observe independent chromosome assortment under the microscope during meiosis. Let's go a little bit deeper into homologous recombination. In 1909, Franz Janssen observed chiasmata, which are points where chromatids make contact and exchange segments during the first meiotic division. This suggests that chromosomes physically exchange segments and alleles become unlinked. Now, synapsis, that's the pairing and interaction of homologous chromosomes, and it leads to recombination. We saw this when we were discussing meiosis. And this is how new allele combinations and new non-parental types are formed. So to illustrate this quickly, let's just consider an example of a heterozygous individual with dominant maternal alleles and recessive paternal alleles. So mom's capital A, capital B, dad is lowercase a, lowercase b, okay? So if the genes are linked, we would expect a 1-to-1 ratio of capital A, capital B, and lowercase a, lowercase b gametes. However, if the genes are not linked, the individual should produce capital A, capital B, capital A, little a, capital B, and little a, little b gametes in equal frequencies instead. So what he did was Morgan discovered that both parental and non-parental cases occurred when test crossing individuals with a homozygous recessive parent. And what this indicates is that while linkage is the majority outcome, a significant portion of offspring resulted from recombination. This figure here illustrates the chiasmata that Janssen observed. And notice these are the same synapses that we saw when we were discussing meiosis in lesson 1. Chromosome isolation and microscopic observation forms the basis of cytogenetics, and it's the primary method by which clinicians detect chromosomal abnormalities in humans. Chromosomal abnormalities during meiosis can lead to inherited disorders. And these abnormalities can be classified into 2 categories. The first one is changes in chromosome number, and the second one is structural rearrangements, and we're going to talk about both of those. Cytogenetic analysis involves isolating and observing chromosomes under a microscope, and it's helped greatly by using a karyotype, or also known as a karyogram. What this does is it provides a visual representation of an individual's chromosomes, including their length, their banding pattern, and the position of the centromeres. Chromosome number disorders are easily identified using a karyogram, and they usually result from duplications or losses of an entire chromosome or a change in the number of sets of chromosomes. We'll go into this in some more detail in just a bit, but I did want to show you figure 13.5. This is a karyotype, or also known as a karyogram, of a female human. So you can see the homologous pairs of chromosomes. They are the same length, the same size. They have the same centromere positions. and they have very similar banding patterns. Okay. You know that this is a female because she has 2 of the X chromosome. If this was a male, it would be an X and then a smaller Y chromosome here. So the chromosome number abnormalities are most easily identified by looking at a karyogram, and it's going to involve duplication of a chromosome or loss of an entire chromosome, and it actually changes the number of chromosome sets that you have in the nucleus. And the main cause of this is non-disjunction. Non-disjunction occurs when the homologous chromosomes or sister chromatids fail to separate during meiosis. Some causes of non-disjunction include misaligned synapsis, an incomplete synapsis, or a dysfunction in the spindle apparatus. And it can happen during meiosis 1 or meiosis 2. If it happens during meiosis 1, then you have 2 gametes that are missing a chromosome and 2 gametes that have an extra chromosome. Conversely, if non-disjunction happens during meiosis 2, then 1 gamete will have a missing chromosome, 2 gametes will have the correct number of chromosomes, and then 1 gamete will have an extra chromosome. And of course, this difference is due to the difference in the 2 cell division cycles, right? So in one cycle, you have chromosomes being pulled apart. In the other cycle, you have chromatids being pulled apart. Figure 13.6 shows how non-disjunction occurs in either meiosis 1 or meiosis 2. So the error is occurring either when a pair of chromosomes fail to separate or when a pair of sister chromatids fail to separate. And we see here in the offspring, we have 2 gametes. And if it's happening during meiosis 1, we have 2 gametes that are missing a chromosome and 2 that have an extra one. And if it's happening in meiosis 2, then we have these 2 gametes have the normal number. But these ones, this guy's missing one and this one has an extra one. Let's talk about ploidy and some of the terminology around it. So we have euploid, aneuploid, and polyploid. And these are all terms that are used to describe different chromosome conditions. Okay. Euploid refers to individuals who have the correct and balanced number of chromosomes for their species. In humans, euploidy corresponds to having 22 pairs of autosomes, which are the non-sex chromosomes, and 1 pair of sex chromosomes. And the total is 46 chromosomes in each cell nucleus. That's euploid. Aneuploid individuals have an abnormal number of chromosomes due to the loss or gain of 1 or more chromosomes. Aneuploidy can occur during cell division when chromosomes fail to separate, or like we just saw when sister chromatids fail to separate properly. Now, I've got a couple examples here of aneuploid associated disorders in humans. Monosomy is a type of aneuploidy where 1 chromosome is missing. So the result is having 45 chromosomes in the cell nuclei instead of the normal 46. Pregnancies with an autosomal monosomy usually end in embryonic death. Now, monosomy of the X chromosome, this is the exception. This is the only non-lethal monosomy. It's called Turner syndrome, and it's a condition that occurs as a result of a missing or sometimes partially missing X chromosome. It causes a variety of medical and developmental problems, including short height, failure of the ovaries to develop, and sometimes even heart defects. Trisomy is another type of aneuploidy where there's an extra chromosome. An example of trisomy in humans is trisomy 21, which is also known as Down syndrome. Down syndrome is a condition in which a person has 3 copies of chromosome 21 instead of the normal 2 copies. This extra copy changes how a baby's body and brain develops, and it causes mild to moderate intellectual and physical disabilities that last a lifetime. Variations in the number of sex chromosomes have relatively mild effects as compared to those we've seen for autosomes. So X inactivation, this is a molecular process in female mammal embryos where 1 X chromosome in each cell becomes dormant and forms what is known as a bar body. This can occur randomly, but females can compensate for it sometimes with their other X chromosome. But if there is no X chromosome present at all, the offspring are not going to develop in utero. Abnormal numbers of X chromosome lead to X inactivation in each cell, resulting in mild intellectual and physical disorders, disabilities, and often sterility. X-chromosomal triplo X, they're XXX individuals. They are phenotypically female, and they may have developmental delays and reduced fertility. Now, XXY individuals, this is called Klinefelter syndrome, and these individuals are phenotypically male, but they have small testes, they have enlarged breasts, and they usually have reduced body hair. More complex types of Klinefelter syndrome may have as many as 5 X chromosomes, with multiple bar bodies in each cell nucleus. Duplications and deletions are another type of chromosomal abnormality that can lead to offspring with abnormalities. Duplicated chromosomal segments may join existing chromosomes, or they might exist independently in the nucleus. Cru-du-Chat syndrome is a notable example where a deletion of most of the small arm of chromosome 5 causes nervous system abnormalities and distinct physical features. Infants with this syndrome emit a high-pitched cry, and that's how it got its name, cry-du-chat. I'm probably saying that wrong. Structural rearrangements of chromosomes, such as inversions and translocations, have been extensively studied. So during meiosis, rearranged chromosomes can pair up with their former homologs to maintain proper gene alignment. However, if the gene orientations on the homologs are incorrect, recombination events can result in the loss or gain of genes, leading to aneuploid gametes. So chromosome inversions involve the detachment, 180-degree rotation, and reinsertion of a chromosomal segment. Inversions can occur naturally due to the mechanical shear or the action of transposable elements. Unless they disrupt gene sequences, inversions usually have milder effects than aneuploid errors. However, changes in gene orientation can affect gene expression, regulation, potentially causing abnormal levels of gene products. Pericentric inversions involve the centromere, while paracentric inversions occur outside of the centromere. Pericentric inversions can alter the relative lengths of the chromosome arms, making them easily identifiable. In inversion heterozygotes, one homologous chromosome undergoes an inversion while the other remains unchanged. During meiosis, one homolog forms a loop and the other molds itself around the loop to maintain synapsis. Although this ensures correct gene alignment, it can result in stretching and imprecise synapsis regions. Figure 1311 helps to illustrate pericentric inversion versus paracentric inversion. Pericentric inversion can change the length of the chromosome arms above and below the centromere. And remember that paracentric inversions do not change the length of the chromosome because they are not happening around the centromere. So just imagine if you have, say, E is twice as long as F, and C is twice as long as D, okay? If you were to invert D and C, then what will happen is this arm of the chromosome will end up being shorter than this arm. Whereas if you were to switch F and E, the length of the arms is unchanged. So that's the main difference between these two types of inversion. Figure 1312 shows the process of inversion pairing. Inversion pairing is that process we were talking about before, where it ensures proper alignment and exchange of genetic material during meiosis in organisms that have a chromosomal inversion. So the process involves the formation of a loop in one of the homologous chromosomes, and this helps it to achieve accurate pairing and successful crossover during the synapsis. Translocations occur when a segment of one chromosome dissociates and attaches to a different non-homologous chromosome. The effects of translocations depend on how gene positions are altered in relation to regulatory sequences. So certain translocations have been associated with cancer and schizophrenia. Reciprocal translocations involve the exchange of chromosome segments between non-homologous chromosomes without either chromosome gaining or losing genetic information. Figure 1313 illustrates the process called reciprocal translocation. It is important to note that this process can lead to new combinations of genes, but It doesn't alter the overall genetic content of the chromosomes that are involved. This concludes the Lesson 6 material. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics, and leave your questions for me in the comments below.